want to really cover today is really aspects of our business, Codex DNA, and really thinking about the idea now of automated, fragment builds, library and clone builds using our novel system, which is the BioXp. Of course, that's my email in front of you, and you're welcome to address any other questions and concerns beyond any commentary following this seminar. So I usually like to start with a specific timeline, really just to contextualize who we are and, importantly, where we've come from and where we're going. Codex DNA is a recent rebrand. We used to be known as SGI DNA, and that was actually a spin out of a lot of the efforts from the Craig Venter Institute. And Craig Venter, of course, as many of you likely know, really helped revolutionize that genomics era, especially with a lot of the whole genome sequencing at the time. But really, we're now in an era of synthetic biology, and really how that came about was some seminal work out of Craig Venter's lab back in the mid-2000s. And Dan Gibson, who's actually our Chief Technology Officer, actually helped inspire a lot of that. So at the time, he actually built the genome of the mycoplasma genitalium. And then a couple of years later, actually went on to really think about synthetics a little bit further by creating the first living cell with an entirely artificial chromosome. Now, what was important about those papers was it heralded in a very novel cloning method. I think cloning up until that point had really relied on the use of, restriction digests and actually creating compatible restriction sites. The use of Gibson assembly completely overhauled that approach, and it really allows the seamless joining of adjacent fragments with the use of overhangs to, again, other fragments. And that, of course, has been utilized in many publications, and certainly in the arena of, for example, immunotherapy, with the generation in this particular paper from Belgium, the generation of nanobody-based chimeric antigen receptors, but of course instrumental in the designs, particularly for CRISPR work, and these are just a few papers from this year. Of course, in the area of T-cell engineering, there's many, many publications, some of which I'll allude to as we go through the talk today. And of course, it's instrumental use in vector and vaccine development, and 2020 has been very key for us in that regard. And of course, mammalian cell engineering, and we have many, many groups, especially across Europe, that do a lot of efforts to that end, especially by applying the aspects of Gibson assembly to that. So again, just to kind of point out some other timelines, of course, we were the first to develop the first synthetic flu vaccine, so that recombinant flu vaccine based on H3N3. And that's really the first demonstration outside of using an egg-based approach, again, to facilitate a very timely intervention from a vaccine perspective. Late in 2015 was when we came out with our first iteration of the platform, the BioXp 3200, which has actually been placed globally now, both in academic, pharma, and contract research associations, again, across many countries globally. And here we are in 2020. Many of us have faced enormous challenges in 2020, certainly having to work remotely from the lab. And really, in part, we've been part of some of the efforts in generating tools for the SARS-CoV-2, and we were able to synthesize the entire genome in just over a week, in part using the BioXp. So it's really now, in 2020, it's about empowering researchers with tools and resources to develop not just vaccines, and it's obviously timely to think about the appropriate vaccine development for SARS-CoV-2, but also therapeutics, diagnostics, and in fact anything really to do with synthetic biology, especially from a sustainability perspective. And we have groups obviously in the ag bio world and thinking about its use for novel biofuels. So again, a lot of innovation really over a very short time period, since the inception of that novel cloning method. But of course, cloning really fundamentally has not changed. And in fact, I even put Gibson assembly as part of this. Most of you will be very familiar with these step-by-step processes that I allude to. Because again, benchtop cloning has not fundamentally changed in the last 30 years. Everyone is still doing what I allude to, which is step 1, cloning. So that's your design through cloning method and transformation. Step 2 is going to be your confirmation, and everybody's still doing colony PCR, mini prepping that up and sequencing. And then of course, step 3 is your expression and scale up. Now, today, that process still takes between a week to 2 weeks. And in fact, that's if you've got very simple cloning. In fact, if you have something like trying to clone in the T-cell receptor or more nuanced cloning that would be associated with, say, antibody builds, that can actually take months and months of work. Again, so this process really needs an overhaul. And really a couple of questions that I do like to posit to researchers is, well, how can you change that? What would your research look like if you actually got your results faster? And I think 2020 really addresses that. There's been certainly a dramatic need to really think about appropriate vaccine development in a timely manner. But importantly, it's about you guys at the bench. So what if you actually had more time that you could devote to other areas of your scientific research problem, and actually you were freed up from doing that very kind of manual processing of your cloning methodologies. And so with that, I do want to introduce to you now our system, the BioXp. Again, it was first rolled out, just around 5 years ago now with our first iteration of the platform, and that's been dramatically overhauled really just this year. And I'm going to talk about our newest system. It really represents a genomic workstation to enable right now your double-stranded DNA applications. And of course, we are thinking of applications even beyond that. So I would say watch this space very closely, because there'll be exciting developments coming your way over the next few months. So it's really about developing rapid and automated applications associated with, perhaps what you've been doing in the past with your more manual benchtop approaches. Again, addressing areas specifically, as I allude to in the cartoon, which is obviously designing antibody libraries, developing molecular tools, and even aspects of cell engineering, particularly with CRISPR or development of CAR-T work, for example. So it's going to save you weeks of that conventional bench work, and for the most part, it is automated, and I'll walk through the workflow with you here in the next few slides. So this is the newest system. We actually just rolled this out in August of this year. It really leverages the latest kind of state-of-the-art software, as well as the state-of-the-art microfluidics. And there's been obviously a lot of work invested in that over the last few years now with the use of high-throughput robotics. And so we've been able to leverage off some of that, again, to enable a much faster system over our current 3200. So right now, it obviously makes synthetic genes in an automated fashion. We can do up to 32 reactions at any given time, meaning 32 fragments, 32 clones, or 32 libraries. Now in the US, it's a 3 to 5 day turnaround. It's slightly longer in Europe, especially when one considers custom and shipping. But our aim right now from a company perspective is to have, from design through final product, everything to be executed within a week, even in Europe. So really right now it's a hybrid of both insourcing as well as outsourcing some of your synthetic DNA needs. And what I mean by that is we will be providing you kits to enable the run on the BioXp, but for example, if you have a very proprietary vector that you do not wish to share, that can be run alongside the instrument. Again, as long as you have designed your vector appropriately, you can come with some of your tools, again, to assemble your final product. And so it's really going to consolidate a lot of those workflows that I alluded to with the kind of traditional approach to cloning. It's really, again, building applications for DNA on the bench with automation and a ready-to-go kit. It's about pushing that button, really, and pressing start. It takes about 5 minutes to load up the instrument, and most people are actually loading it up about 5 o'clock in the afternoon. And so while you're away from the lab, in fact, in an overnight run, it enables obviously the assembly of your fragment, clone, or library build. So right now the instrument, the 3250, is very versatile. We have it IQ, OQ'd, and PQ'd, and we are working a path for deployment from a GMP perspective. So right now it can be deployed in both research or clinical settings. And of course it's going to drive your productivity in a lab quite significantly, especially because it can free you up from doing a lot of those manual approaches associated with the current paradigm for cloning methodologies. I keep mentioning that we do have IP already built in. So that vector, again, that proprietary vector, and this is especially important if you work in vaccine development, it never actually has to leave your lab. And so this is the current workflow. Again, one would submit your sequences through our online secure portal, and we've just overhauled that portal with the latest state-of-the-art bioinformatics. So you will get real-time feedback on the complexity of your sequence. That sequence can be uploaded as a GenBank or a FASTA file. And we will actually give you tips and tricks on even improving that design. Again, all in real time. If your sequence is graded correctly, you will receive your entire kit for that. So for example, your fragment builds come in what's called an oligo vault. Fragments are actually built from much shorter oligos to assemble the much longer fragment that you wish to clone into your backbone, for example. Again, you're receiving your entire kit from us. You will load up the system, and really it's about walking away. The actual protocol is very specific to you. We have a QR code that is designated on our oligo vault, and the camera in the system will recognize that and again pull that specific protocol for your run. It's an overnight run, so the next morning you retrieve your synthetic DNA, which again can be either fragments, clones, or libraries. So as far as applications, I keep alluding to the fact that it is fragments, clones, and libraries. With fragments, you can build individually up to 1,800 base pairs. Of course, that's not to say you can't do 1,900. It's not to say you can't do 2,000. But we do actually have what's called a rule of engagement for a Gibson assembly, which I'll allude to in the next couple of slides. You can actually stitch together several of those fragments up to a total of 7 kb, and you can clone that into any backbone vector in so much as you've appropriately designed. And of course, you can build your custom variant libraries, and I'll allude to several examples of that during the talk. So these are the kits. Again, you can build, as I keep reiterating, fragments, clones, or libraries, and your clones can be single insert or multiple insert. You can also elect to do what's called an amplification of those clones. We include actually a master mix for that if you wish to amplify, and the benefit of that is that you can bypass the transformation process entirely. So while I said you can do 32 reactions, you don't have to do 32. The robot actually processes 8 at a time. So one can do as few as 8 samples. So you can do 8, 16, 24, or 32. Now, if, for example, you don't want to use your own vector, you'd rather have us actually provide you backbone vectors, and then you can subclone out if you desire. We do have a portfolio of backbone vectors with different resistance markers on them, and we will continue to increase our portfolio of these vectors. They come already designed, they already have the necessary overhangs to facilitate an automated cloning on the instrument. So please feel free to go to our website and take a look at some of those. So I'm just going to go through some of the specifications for fragment and clone and library builds. So I did mention that there is kind of these rules of engagement for a successful Gibson assembly, and with that our sequence guidelines are between 300 and 1,800. Of course, you can really go up to about 2 kb, you know, without any problem at all. But again, it's kind of proceed at your own risk. We've certainly validated the 300 to 1,800 quite substantially. Of course, there's limits with regard to GC content. You don't want to go over 70%. And of course, we will have an algorithm that also incorporates anything such as secondary structure issues and tandem repeats. One of the benefits of running automation over and above, for example, a benchtop approach, is that we include a very novel 2-step error correction process. It's actually a mismatched nuclease and a high fidelity polymerase that's actually included in the automation. Again, over and above what you'd actually see at the bench. So that ensures the fidelity of your product. And again, it's an overnight run achieved on the instrument. Now, with regard to clones, there's flexible options. One can do single fragment cloning, such as I allude to in the cartoon, or you can do multiple fragment cloning, meaning that you can stitch up to several of these fragments, again, into your backbone vector. It can be one of ours, or it could be something that you provide. If you remember, the robot does 8 samples at a time. So some people actually elect to think about, well, what's the most appropriate backbone vector for my purposes? And so you can actually, in a single run, interrogate up to 4 unique vectors, again, as part of that. Again, the benefit of cloning is that not only do you receive your clone product from the instrument, you also receive the fragments that go along with that. So we will build the fragments and we will build the clones. So you get the flexibility of both products, again, after your run. So just to kind of touch on multi-fragment, again, it's the ability to really stitch together several of these fragments. Again, right now it's up to 7 kb. This will likely change over the course of the year as we aim to kind of improve our processing. And again, that can all be cloned into your backbone vector. So you can actually include an optional constant region between your fragments. And certainly I allude to that in the cartoon also. And it's certainly approach for kind of antibody builds and novel antibody builds using the multi-fragment approach. So really you can think about assembling 6 pieces. And what I mean by that is your backbone, your fragments, and your constant region. So it does allow you to really generate some of these larger constructs or even single chain variants in as few as 5 days, and especially in an overnight run. And again, to reiterate, you'll receive both your cloned and uncloned products back. So just kind of to summarize some of that, certainly for fragments and clones, it really enables a really fast turnaround time, especially when one thinks about antibody development, you know, and efforts towards, you know, vaccines, for example, significantly faster by doing it in an overnight run. And what we rolled out earlier this year was our error-free cloning guarantee. Again, the devil in the details is always going to be in your upfront design. If you can come to us with your sequences and we work through your design attributes, and again, the algorithm grades that as appropriate, we will guarantee you success in achieving your error-free clone. And that's something that, for example, Celebrate in the US came to us with. They actually had several insert sizes into their backbone. And they ended up picking 56 total clones. Only one of those actually had an issue. So the error rate for that was 1 in 83,000 base pairs. So how that all plays out mathematically is you've got an average error-free cloning of 98%. Nothing's ever going to be 100% in science, and I wouldn't expect it to be so. But we do aim to have at least over 95%. And again, most of our customers do not pick many clones. In fact, we do suggest picking about between 2 and 4 clones for sequencing. And this was an approach that Autolus came to us with. Autolus is a pharma company in the UK. And obviously they're assembling some of these single chain variant sequences, and you know, this is their traditional workflow. This is probably very familiar to you because this is something that you're likely doing right now, going through the design process, again, filling out your details, generating those fragments, assembling, ligating, colony picking, and sequencing. Well, we overhauled that entire process using the BioXp, again, in an overnight run. And importantly for the customer, it was really about the reduced error rate of assembly. But overall, you're really spending less time assembling your constructs, and that in itself leads to much lower costs and actually a higher capacity of their output and workflow, which you can actually see here if you hone in on the middle figure, where you can compare the kind of build, design, test cycle paradigm. And of course, it's significantly longer with that traditional approach that I allude to. And obviously, you're really limiting the time spent by doing an overnight run on the BioXp. So that in itself will increase the capacity of your pipeline, in this case by over 25%, allowing that valuable resources to be available for the downstream validation and testing. So I'll just touch on the rapid amp, again, the ability now to amplify your clone, again, in an automated fashion, because really the benefit of that is to eliminate, again, that process of downstream transformation, growing and prepping. Again, you're achieving a very high yield with that amplification using the rapid amp master mix over and above that kind of traditional methodology of cloning. And so really it's about achieving, you know, everything from fragment all the way through to transfection ready DNA in an overnight run. So obviously it makes your fragments, you can clone into your backbone vector, and you can really scale well beyond 10 micrograms. In fact, we have customers that have achieved well over 50 micrograms, again, in an overnight run through automation. So again, the benefit of this is to kind of skip that E. coli transformation, and again, avoiding any potential endotoxin contamination. So in a kind of seamless workflow, it's all about generating both assembly and amplification. And how the process works, of course, is to start off with a circular template. We have random hexamers that bind to that. There's a very specific polymerase that recognizes those as primers. Polymerization begins and you get strand displacement. So this is really evidence of rolling circle amplification. So each of these new strands then become templates for the polymerization, and it actually keeps occurring around the original circular template, as you can see here. So what's generated off the instrument are these highly structured concatenarized products. So what then one has to do is to take that product, do a restriction digest and linearize that, and that linear template can now be directly transfected into your host cells. So this was an approach that several of our customers have been doing. In fact, we've had customers right now that have adopted this as part of their vaccine development for SARS-CoV-2. I look forward to some very exciting publications here in the next month or so. But we did have a very recent study whereby the customer had 3 fragments, again, all under the guise of our sequence considerations, coupled to a fusion tag. And we were actually able to generate not just the assembly, but also amplify that and linearize that all in an overnight run. So I'm now going to kind of change tact here a little bit. I'm moving on from you know, gene fragments as well as cloning now to think specifically about libraries, because we've actually had a lot of publications really in the kind of last few weeks, especially in libraries, and especially with the design of novel antibodies using epitope mapping strategies and scanning library strategies to look for novel antibodies for the SARS-CoV-2. In fact, there was a recent paper addressing antibodies to the receptor binding domain that interacts with the spike protein of SARS-CoV-2. And again, they did that with the use of a scanning library using the BioXp. So I just kind of give you the nomenclature of that and what each of them mean. But again, if you're interested in libraries, we have a lot of publications to that end. And again, just some of the applications that people are using these libraries for, especially right now in epitope mapping and of course antibody engineering and protein engineering. Now, a recent paper actually out of Boston in the UK kind of addressed the use of some of these scanning libraries. And I'll just kind of briefly go through really the issue at hand and the publication that came from this work, and it was published in Cell earlier this year. So the group actually are investigating a toxic protein that's made by a bacteria that when it's initially expressed, it forms a very tight disc. Now that protein is secreted by the bacteria inside the cell. When it gets taken up into the lysozyme, what you get is this change in pH, and that now causes that disc to form a spike that actually bursts the lysozyme, and that kills the cell. So now you can start thinking about identifying these residues that actually cause that and perhaps modulate this behavior of the protein for containment purposes. So how do you go about that? When you don't know what you're looking for, of course, and you don't know the residues, sometimes a good approach is to do what's called this NNN site saturation, and in this case, that's what the group did. And so they designed a 436 base pair construct with this NNN site saturation. I allude to that, you know, in the diagram here on the right-hand side. So they're actually sending us, obviously, their sequences on a Monday, and we're able to synthesize all of these scanning libraries by Friday, and it really allows you to really quickly identify the functional residues. Now, with libraries, it's much quicker. It's actually an 8-hour run for 32 of these, because there's a level of degeneracy actually built into that. So, for a lot of customers, you can do 96 reactions in that 24-hour time period. Now, we do get asked a lot of questions about diversity, and this is just an example from that paper whereby all those fragments that were generated off the BioXp, we actually put them into a PacBio sequencing, and from the 64,000 reads, we're actually able to get the true level of diversity. So what you're seeing here in that figure on the right-hand side is on the top, you've actually got representation from all 20 amino acids, and then on the y-axis is all the 104 amino acids that we actually designed for. So the dark blue are all the mutations that we introduced, and so you're specifically seeing the changes that are made, and in the light blue are all the wild types of that particular position, and the oranges are actually the misses. And so really from a single sequencing reaction, you get this great data, and it's more than 95% coverage, and you can actually see kind of all the possible combinations and the true level of diversity that is achieved. Now, this is a recent library build from Belgium, whereby they're developing what's called a nanobody library. Nanobodies are actually, getting a lot of social media coverage right now for their kind of novel therapeutic aspects, and certainly one of the first papers attesting to that was from BIB actually in Belgium. So I kind of do a compare and contrast with their manual workflow. Again, that should be very familiar to you if you are cloning at the bench. And again, it's a long process to do this because you've got to kind of independently process here your variable sequences and your FC region, and then finally ligate into your destination vector. You can do all of that really in a week's time using the BioXp, and again 100% of the customers' constructs could actually be synthesized and cloned directly into their expression vector in an overnight run. So again, significant time savings, and it does allow then that molecular biologist to focus on other pertinent areas of the scientific question and goals of the lab. And so this was the paper that came out of that. Again, it's really kind of interrogating a nanobody-based library as kind of novel carriers for targeted vaccine delivery, in this case designing potentially oral vaccines. And of course, I pulled this directly out of the publication addressing their specific methodology. And one thing at the time, and this was last year when a lot of this was run before we had the two-step error correction. In fact, when the customers actually picked just 2 clones, 97% of them were error-free. This is a recent publication addressing to really think about how does one go about identifying clinically relevant T-cell receptors. And that actually takes a lot of work. There's been a lot of bottlenecks in this area for a long time, and one of our customers at Gigammune Pharmaceuticals in San Francisco has recently been adopting the BioXp to address aspects of that. So the goal of this paper was to integrate the BioXp with other high throughput methodologies. And you can see aspects of that. Now, I don't want your eyes kind of focusing all over the place. Certainly, if you look at the right-hand side kind of methodology, it's really about, starting off with these droplet microfluidics, again, to natively pair the alpha-beta chain of the TCR. And then now we can integrate the BioXp, and that's highlighted here in the center figure. This was all done in an automated fashion using our initial system, the BioXp 3200. Again, in an overnight run, you can generate that product, which you can then package into lentiviral particles and transduce into JERCAT T cells, creating a library that you can then subsequently do some deep TCR sequencing and perhaps follow up with validation and activation screens, and specifically T cell mediated activation screens. And so that really got a lot of press. There was a lot of buzz in the kind of social media on this, because it really kind of allows one to kind of very quickly identify, in this case, 10 to 100 or more natively paired TCR alpha and beta sequences. And really kind of it absolutely removed a substantial bottleneck in kind of thinking about T-cell receptor discovery, but also importantly, how one goes about the development of new adoptive cell therapies. I'll just touch on some of our efforts, specifically with regard to how we went about for the kind of SARS-CoV-2 strategy. Obviously, I allude to our process on the left-hand side, and I kind of highlight aspects of that here on the right-hand side. So, in the last kind of few slides, I'm just going to walk through obviously the portfolio of products that we have really come about with over the last few months, and it's really designed to accelerate the development of solutions for COVID-19. Now, the first of which was actually the development of the whole genome. We actually produced that entire genome in under a week, in part using the BioXp. Now, we did this in 3 stages. Now, stage 1 involved the BioXp generating 24 1.4 kb fragments. And again, that's all done in an overnight run. Stage 2 is actually transferring that to the benchtop, whereby we did benchtop Gibson assembly and combined these stage 1 fragments into stage 2 fragments. And then these stage 2 fragments were further assembled into the full genome of the SARS-CoV-2. Again, using benchtop Gibson, and we cloned into a back. Now, you want to validate that you've actually generated the right product of the BioXp, and certainly we encourage customers to run a gel. Now, in this case, we also verified, since we're commercially promoting a product, that sequence is correct, and so we use Sanger sequencing. And just some further validation, you certainly want to make sure that you have assembled into the full length, in this case using Gibson assembly cloned into the back, so we use field immersion gel electrophoresis as well as supercoiled DNA analysis. Now, since that time, and that was developed in March, obviously at the kind of height of the pandemic, we've been able to develop a full repertoire of tools, obviously using the RNA controls. And actually the development of the RNA controls has led us to think about developing RNA applications for the BioXp. We've expanded our comprehensive research tools, especially as we now know more about the specific spike protein mutation, the D614G mutation. Obviously having expression ready constructs for that, and as well as tools for vaccine research with panels for the antigen panels and obviously spike protein. So again, to really kind of summarize, it's about 2 things really. You don't have to look at all these bullet points, but it's really about the fast turnaround time over conventional methods, but importantly it's about the high quality of the build, again in just an overnight run. And so we really think about impacting scientific research, again, with automation. It's not just about, as you see on the right-hand side, which is vaccine development and genome engineering. Of course, we were able to build the genome for the SARS-CoV-2 in really an overnight run on the BioXp, and we continue to work on genomes. We've certainly got a lot of customers interested in other viral genomes, and it's something that we do very well and we've been doing for a long time. So this really lends itself to dramatic improvement in lead optimization. But of course, it's in areas such as biologic drug discovery, and you saw obviously some of the results that you can get just in that overnight run with DNA libraries. Again, all about improving your lead optimization productivity, but also in personalized medicine and thinking about strategies in immunotherapy. And certainly one of our key customers that publishes routinely with us is Steve Schoenberger, out of the Immunology Institute in La Jolla, and he certainly has, it's enabled a lot of his research, especially in T-cell and T-cell receptor work using the BioXp. So I'll end here because I know many of you are used to thinking about sequences and used to thinking about reading DNA, and you've probably got the capabilities of Illumina, and Illumina of course are actually right around the corner from us in San Diego, so we kind of benefit a lot from some of the groups actually at Illumina. But reading DNA is really only one part of the kind of full picture and big picture associated with the sequences. one has to have the ability of writing that DNA as well. So, you know, we like to think of this as a kind of very unique marriage of both reading and writing DNA. And again, many of you already have the capabilities from a sequencing perspective. But again, that only tells one aspect of the story. You've got to be able to write that. So again, I thank everybody for your time. Again, that is our website, codexdna.com. I did allude to my email in the very beginning, and I'm more than happy to take any questions. Thank you.